All right, that means go. So, welcome again, everyone, to session 4. My name is Dr. Bill Frank. If you have not been here before, I am going to serve again as your humble moderator for this evening. So, I have the pleasure of announcing our presenters this evening. And our first presenter for anatomy of the nervous system is Dylan Persaud from Drexel University College of Medicine. Dylan is from Long Island, New York, and graduated from Hofstra University in 2022 with a major in biochemistry. He is currently a second-year medical student at Drexel University. In his free time, he likes to work out, spend time with friends, and play basketball. Academically, he is interested in pathophysiology, anatomy, and pathology, and wants to specialize in pulmonary critical care medicine, emergency medicine, or trauma surgery. So with that, the floor is yours, Dylan.
Okay, thank you, Dr. Frank. I'm just going to share my screen now, and then we'll get started. Okay, and you guys can all see my screen, right? Okay, great. Just going to get my screen sorted. Okay, good evening, everybody. So as Dr. Frank mentioned, my name is Dylan. We're going to be talking about the gross anatomy of the nervous system. And I also have, some references in the bottom left corner of my screen from previous tutors who I've used inspiration from their presentations to create this one. So the first thing we're going to do is just go over the objectives. So you guys can read this, in your free time, but just very briefly, we're going to go over the anatomical and the functional aspects of the nervous system, talk about some different classifications and also the function, so central versus peripheral, sensory and motor, talk about the lobes of the human brain and the different functions of the lobes, differentiate between the somatic and autonomic nervous system and their functions. We'll talk a little bit more about the granular aspect of the spinal cord, so more of the nitty gritty, the white matter, gray matter, parts of the spinal cord, and then just wrap it up talking about the 5 special senses, which I'm sure we're all comfortable with, but talk a little bit more about the anatomical side and the neuroscience side of that. And I have a link for the tutoring session from last year that you guys may have reviewed. So we're going to start off with the questions. So Dr. Frank, if you could just do the poll for this. So which of the following statements correctly differentiates the central nervous system, or the CNS, from the peripheral nervous system, or the PNS?
And just jumping on, if this is your first time being with us, when we have questions that are this long as far as the reading, we'll give you some extra time. So take the time that you need to select an answer. And we're looking for about 75% of you, at the very least, to have an answer in before Dylan starts to review the correct answer with you.
And we'll definitely be able to talk about it. You guys can ask questions, so no pressure.
We're going to end the poll in 3, 2, 1.
Okay. All right, choice number 2. Great. So that is correct. So the CNS does include the cerebrum and the spinal cord, and the PNS includes the nerves and the ganglia. I know that I think choice A or choice 1 was the other one that was also chosen. Almost there. It's just a little bit switched around. Yes, we had like about 15% for choice A, which, that's just maybe the pressure of the first Zoom question of the night, which is okay. So the CNS versus the PNS. So the central nervous system versus the peripheral nervous system. I have a picture here. So the central nervous system consists of the brain and the spinal cord. And the way I like to think about it is that everything that's not the brain and the spinal cord is the peripheral nervous system. So again, that's the brain or the cerebrum and the spinal spinal cord is the central nervous system. And then we have ganglion, nerves, etc. That's part of the peripheral nervous system. So your peripheral nervous system is kind of the component of your nervous system that's doing kind of like the heavy lifting in terms of actually like, picking up and grabbing stuff or sensing things. And your brain and your spinal cord is kind of like you could think of your spinal cord as like a relay station, and your brain is where all of that information is kind of getting interpreted. And we'll go into this a little bit more, talking about, each of these aspects and what they actually do. Okay, so the next question, which of the following statements correctly describes the function of the axon of a neuron?
Ending the poll in 3, 2, 1.
Okay, so this one we got a little bit of a spread. It looks like choice D and choice A were the most chosen. So this one is actually choice D. So there's a little bit of nuance here, but we can spend some time going over this because we had a little bit of a spread. So the axon is the part of the neuron that is carrying the information away from the cell body. And I have a couple of pictures that we can go over and use to illustrate this. But choice D is correct. It's carrying away the electrical signals from the cell body towards the next part in the circuit. So that might be a neuron, that could be a muscle. Okay, I know that A was the second most chosen choice. So it receives signals from other neurons and transmits them to the cell body. That would be your dendrites. So that's a little bit closer to the cell body. It's kind of coming off as little tree branches, but its long kind of tail, you can think of it, is the axon, and that's what it's doing. It's carrying away the electrical signals. I know B and C were also chosen. So for B, it doesn't store ions in the synaptic vesicles, but it actually stores neurotransmitters. And for C, the part that insulates the neuron or insulates the axon more precisely is the myelin sheath. So depending on your objectives, you'll kind of know what's important, but I'm just going to move forward and talk a little bit more about those parts of the question. So like I said, we have our cell body or our soma, and what's receiving, you can kind of see it's a little bit faded here from the previous axon. Those are our dendrites. So that's what's receiving the signal. Then it's going to get assimilated in the cell body and then carried away by the axon. And then you could see we have a synapse here, and towards the bottom, we have the next neuron in the circuit. Okay, so we have the axon, the myelin sheath is what's insulating it and protecting it so that the electrical signal moves smoothly to the next part of the circuit, whether that's a muscle or a neuron. And here we have just a zoomed in portion of the synapse. So you can see in the axon terminal and the synaptic vesicles, these are actually neurotransmitters that are in here. And there is some involvement with ions, which we won't really get into, but just know that in the synaptic vesicles, we have our neurotransmitters. That's what's actually being released. And then it will be released into the synapse where it can then bind to the target or the effector cell. Again, that would be a neuron or a muscle in this case. Okay, so I hope that is a little bit more clear. So we have different neuronal cell types. They come in different flavors. Okay, so we have unipolar, we have bipolar, pseudo-unipolar, and multipolar. So unipolar has one process or one axon, and it doesn't have anything else coming off of it. It just has a myelin sheath that has one axon, and then we'll have its processes down here. Again, you could imagine that this is where your next neuron or muscle is going to be. Then for bipolar, I like to think of it as having two arms. So you have some sort of receptor in the periphery, like in your skin, which again, we'll talk a little bit more about that later. So we have a sensory receptor that's going to take the information towards the cell body, and then it's going to use its axon, the remaining part, to then transmit that information. Okay, so that's the bipolar neuron. Our pseudo-unipolar is typically associated with sensation as well. So we have sensory receptors, and then it's going to travel from the direction of the sensory receptors towards its other process while being assimilated in the cell body. So it's going to take all those signals and then it's going to be processed by the cell body and then transmitted to another neuron. Okay, and then the multipolar, I like to think of MM as motor. So these are the neurons that are specifically going to be acting on your muscle cells. So I like to think MM, so multipolar is motor, and the pseudo-unipolar, so the S sound is for sensory. Okay. So now we'll talk a little bit more about gray matter versus white matter. So white matter is on the inner part of your brain, whereas the gray matter is more so on the outside. And it's actually flipped when you compare it to the spinal cord. So we have white matter on the outside and gray matter on the inside. So the gray matter is actually collections of neuronal cell bodies. So remember, if we can go back, yeah. So if we can go back, so the cell bodies that are all clustered together, when those are together, it forms the gray matter. And the gray matter, depending on if you're in the CNS or the PNS or the central versus peripheral nervous system, the central nervous system, those collections of our cell bodies are going to be called nuclei. Whereas in the peripheral nervous system, they're going to be called ganglia. And again, I have some more pictures to kind of reinforce this later in the presentation. But just know that if it's a collection of cell bodies, it's going to be gray matter. And that gray matter can then be classified as a nucleus or a ganglion. And that depends whether it's CNS, so remember cerebrum / brain or spinal cord, versus peripheral nervous system, which is our ganglion, our nerves, etc. Okay, so now our white matter, we already talked about how our cell bodies are our gray matter. So the white matter is actually our axons, which is our myelin sheath. So if you were to ever go into a dissection class and look at the nerves, they actually have a little bit of like a yellow whitish tint. And that's actually partially because of the myelin, which is a fatty substance, which kind of looks yellow to white, which is why it's called the white matter. So that white matter consists of our axons of our neurons. They can also be called tracts, TRACTS. So if you see that somewhere, you'll know that it's also white matter. And again, those are bundles of axons. So think of them kind of like a bundle of wires that are transmitting an electrical signal from one place to another place. So that white matter is our cables of our axons, and they have a common origin and a common destination. So the tracts have a common function. Okay, and in the peripheral nervous system, these bundles are actually called nerves. So the nerves that innervate your skin, innervate your muscles, those are white matter. That's considered white matter. So, and the gray matter and the white matter is in the cerebrum as well as the spinal cord, as we have in these pictures up here. Okay.
Hey, Dylan,
And
it's Dr. Peterson. Before we move on, I just wanted to kind of give a shout out to Kenzie, that we have a couple of questions in the chat about these different sort of morphologically shaped neurons and why that matters functionally and how to identify them. So I'm just going to say on the recording that Kenzie is going to cover that material. Dylan should probably just keep going since that's going to be a little bit reinforced with her presentation and he still has a lot of information to cover.
yeah, so that'll be covered more in the micro section, but thank you guys for asking those questions. Okay. All right, so now talking a little bit more about the spinal cord as we move along. So we have different parts of our spinal cord. We have the spinal nerves, and then we have the roots. Okay, so the spinal nerves are bundles of axons that come off of the spinal cord. So you can see it would be corresponding to this portion, and it contains sensory as well as motor axons. So you can see the sensory portion are these blue fibers, and the motor portion are these red fibers. So when they come together, they form the spinal nerve. And we actually have 31 pairs of spinal nerves. This is a fun fact. And our roots are closer to the spinal cord, but they actually only have one of each kind of functional separation. So the dorsal root is only sensory information, whereas the ventral root is only motor information. So you can see here that even though the spinal nerve contains both, the sensory information is going to pass through what we call the dorsal root ganglion, which is our pseudo-unipolar neuron. And then that information will then get put into the spinal cord via the dorsal root. So the roots only have one kind of information. As Dr. Peterson likes to say, it's a one-way highway. So the roots are a one-way highway. So it's only one going in or one going out. Whereas our nerves are two-way highways. So we have sensory information going in, motor information going out. Okay. So for the next question, you accidentally touch a hot stove and quickly pull your hand away. Which pathway correctly describes how this information travels the spinal cord through this reflex? So remember, we talked about one-way highways, what's sensory, what's motor.
Ending the poll in 3, 2, 1.
Okay, choice number 2. Great. So that is the correct answer. So remember how I said we have our one-way highways? So the one-way highways are our roots. So we have the sensory information going through the dorsal root, and then that's going to be processed in the spinal cord and then, or in the brain, depending on what kind of information it is. And then through the ventral root, we have our motor signals. So that would be what would happen if you touched a hot stove, okay? Because that's a reflex. So I'm just going to make this picture a little bit bigger just to talk about it. So now we have sensory and motor components of the spinal cord. So I kind of just talked a little bit about it, about our one-way and two-way highways. So we have our two-way highway. This is our spinal cord here. One-way, which is our sensory because it's our dorsal root. So dorsal is sensory. And then our ventral portion, which is going to be motor. And that's going to be our ventral root there, spinal nerve. Okay, so once we have some sort of stimuli, so let's say painful stimuli, we're activating our nociceptors. And those nociceptors are specific, their job is to sense pain. And that sensation is going to travel, as we see in this graphic, through the dorsal root ganglion, through the dorsal root. Sorry, I think this light is shutting off on me. But through the dorsal root, and then it's going to get interpreted by the or assimilated by the spinal cord because it's a reflex. So reflexes just travel through the spinal cord and through our nerves. Okay, and this is the afferent signal with an A. Okay, so I like to think of the A as arriving. So this signal is arriving to the spinal cord. So it transmits the signals to the corresponding motor neurons. So you have specific nociceptors and sensory neurons in your arm, finger, and those same motor neurons are going to get activated so that you can pull your hand away. So the motor neuron is going to send signals to that muscle to move away from the painful stimulus. And that's going to be via the ventral root, and this is called our efferent signal. Okay, so just remember that the A in afferent is arriving towards your body, towards your CNS, and the efferent is what's actually doing the work, so pulling your hand away. So for the next question, which of the following correctly matches a division of the nervous system with its primary function?
Ending the poll in 3, 2, 1.
Okay, choice 4 was our most common answer, and that is correct. So remember, our parasympathetic nervous system is our rest and digest. And just to talk a little bit more about each of these choices, so the sympathetic nervous system would be what we can call our fight or flight. So that's going to ramp up body activity, increase your heart rate, increase breathing, things like that. The autonomic nervous system versus the somatic nervous system, so these should actually be flipped. So the autonomic nervous system is involuntary control, whereas the somatic nervous system is your voluntary control, okay? And here we just have divisions of our CNS and PNS. So just a reminder, CNS, central nervous system, brain and spinal cord, peripheral nervous system, consists of everything that's not that. And we can actually break up the peripheral nervous system into more categories. So we have our somatic nervous system, which is our voluntary. So, if I were to pick up a pen or scratch my head, that's all somatic. Whereas autonomic nervous system is involuntary. So more so thinking like control your heart rate. I can't tell myself, okay, I'm going to speed up my heart rate right now, right? So things that you can't necessarily control. So just think about it like that. And then from there, we have our sympathetic and parasympathetic. So sympathetic, fight or flight, parasympathetic is rest and digest. It's a good way to think about it. And just some more details. So we have the somatic nervous system, again, which is our voluntary action. That's our somatic motor and sensory fibers. So what we were talking about before, when you touch the hot stove, that's all part of somatic nervous system. Whereas our autonomic nervous system is our involuntary functions and consists of autonomic motor fibers. So again, like I said, we have fight or flight for sympathetic. So heart rate increases, we can slow digestion, we produce adrenaline so that we can, run away from a threat or fight a threat. Our parasympathetic nervous system is our rest and digest, so kind of the opposite of sympathetic. And then we also have another division called our enteric nervous system, which is basically the part that controls our GI tract, that controls movement of your smooth muscle of your GI tracts to kind of promote digestion. So another question, which lobe of the brain is primarily responsible for executive functions such as planning, reasoning, and problem solving?
Yes, second here. You end the poll in 3, 2, 1.
Okay, great. So we have frontal lobe as our leading answer, and that is the correct answer. I'm just going to advance the slide because we'll be talking a little bit about it here. So we have different lobes of our cerebrum or our brain. So the frontal lobe, as we just talked about, or in the question, a lot of you guys got it right. It's what we call executive function. So that has to do with planning, reasoning, problem solving. And this is actually, I'm now seeing a picture of Phineas Gage. So if any of you guys are interested, that's a very interesting case report about the functions of the brain and the frontal lobe. And before we get into, kind of the stuff that's going on up here, I'm just going to move to the parietal lobe. So the parietal lobe is primarily sensory. So we're talking about pain and temperature sensation, 2-point discrimination. So if I pinch my finger versus the top of my hand, I can kind of tell where those 2 points are. And also spatial relationships. Our temporal lobe down here is language and speech and forming memories. So that's kind of closer to like your ears. So I like to think about like, things that you're hearing. So language and speech, and also forming memories if you hear things and remember them. And your occipital lobe is for pretty much vision, depth, identity of objects, space, shapes, where they are in kind of your visual field. And then up top here, you could see for our frontal lobe and our parietal lobe, we have a couple of different divisions here. So still within our frontal lobe, but more towards the front, is going to be our motor cortex. So this is called our primary motor cortex, which is responsible for coordinating our movements. So all of the information that goes to our brain that has to do with motion, it's being kind of controlled with the motor cortex. And the somatosensory cortex is directly behind that and is in our parietal lobe. So the frontal lobe, I like to think of action, and the parietal lobe of sensation. So the frontal lobe corresponds with the motor cortex, and then the parietal lobe corresponds with the somatosensory cortex. So somato just means somatic, and sensory is sensation, okay? And the brainstem down here has to do with basic life functions, breathing, heart rate, and things like that. It's responsible for very basal functioning of the human body, and it connects the cerebrum to the spinal cord, okay? And our cerebellum also, I missed that, has to do with coordination, posture, and balance. And yeah, that's pretty much it for the cerebellum. So which structure of the eye is responsible for detecting light and converting it into neural signals for the brain to interpret as vision? So specifically detecting the light and converting it to signals.
Mm. And the poll in 3, 2, 1.
Great. So large portion, about 80% of you guys got it right. So that would be the retina. So the retina is actually what's doing the integration of in terms of processing the information from light that's hitting your eye. And these other parts do play a part in, the functioning of your eye, but not necessarily the conduction of information. Okay, so now we're going to get into our special senses, and this is kind of like the last large segment of our talk. So we have basic structures of the eye. So we have the sclera, which is the white of the eye, and we have the cornea, which is allowing the part that allows light to enter. The choroid is what supplies blood to the eyeball. So that's the vascular supply, the blood supply to the eye, which is very important. The ciliary body is a muscular structure that allows the lens, which is another part of the eye, to accommodate. So basically, nearsightedness versus farsightedness, and focusing your eyes on like a close computer screen versus you're trying to focus on something far away. And the lens is the transparent structure that focuses the light to the back of the eye, which, like I said, the ciliary body is kind of helping with that. The iris is the colored part of the eye, and it also has a smooth muscle portion that opens and closes the pupil. So that would be dilation and constriction of the eye. The pupil is the hole at which the light enters, and then the retina is the innermost layer towards the back here. That is part of your optic nerve, which is actually processing the light that's hitting the retina and then taking it back to your brain. Okay, and then obviously the optic nerve is carrying that visual signal to the occipital lobe of the brain. Okay. What is the main function of the cochlea in the inner ear?
Okay. Ending the poll in 3, 2, 1.
Okay, great. So most of you guys got this one right. It's converting sound waves into neural signals, and we'll talk about that here. So sound waves enter through the large flesh structure of the ear, which is the auricle, so pretty much the visible ear, and then travels down the auditory canal, the ear canal, and vibrates the tympanic membrane. And we have very, very small bones, which are called ossicles, that also vibrate and cause the bone to brush against the inner structure, the structures of the inner ear, which is the cochlea, which allows for sound to be transmitted. And we also have another part of the ear called the vestibule, which is responsible for balance. And the cochlear nerve and the vestibular nerve is what is transmitting this information to the brain. And they're actually grouped together as one cranial nerve. Okay. So a student accidentally burns their tongue and damages several of their papillae. As a result, they have difficulty detecting sweet and salty foods. Which structure within the papillae would most likely be affected?
Ending the poll in 3, 2, 1.
Okay, so for this one, it looks like we had like a spread between A, C, and D. So taste hairs aren't a structure that we're talking about here. And we also have oh, I think the answer actually got changed from when I went back. But regardless, we'll talk about it here. So the tongue has raised bumps called papillae, which contain our taste buds. So that would be differentiating between salty and sweet, acidic, etc. And the taste buds have specialized receptors that bind to the corresponding chemical in the food. So salt binds to sodium. Acidic would be for H+, sweet would be glucose, sucrose, etc. Okay, and these signals are passed onto the major nerves that innervate the many areas of the brain, allowing us to perceive taste. Okay, and these are 5 basic tastes. So that would be sweet, sour, salty, bitter, and umami. Um, and that's pretty much it for the tongue. Which of the following correctly describes the pathway that smell information takes from detection to interpretation?
Ending the poll in 3, 2, 1.
Okay, great. So most of you guys got this one right. So the odor molecules are going to get interpreted by the olfactory receptor cells, which are going to take into the olfactory bulb and then assimilated as information at the brain. So when breathing, chemicals in the air travel into your nasal cavity. So you can see in this picture here where it contacts the mucus and the muscles, and we have the pathway here. So the sensory receptors are going to transmit that information to the olfactory bulb and tract. And the olfactory nerve is going to take the signal of the smell molecules to many parts of the brain. So sometimes it can stimulate, obviously, interpretation. So what are you smelling? But it can also bring back memories and things like that. So it interacts a little bit with your parietal lobe. And the olfactory nerve travels through the creepiform plate of the skull through the tiny holes in the nasal cavity. And when the olfactory nerve transmits the signals to the limbic system, different smells can be associated with emotional memories because our limbic system is associated with emotion. Okay. So like I said, emotion with the limbic system, it could also bring back just like certain memories just in general through the parietal lobe as well. Which receptor type allows your body to sense the stretch of a muscle when you begin to lift a heavy object?
Ending the poll in 3, 2, 1.
Okay, so it looks like A and C were the most chosen choices, and we're going to go over that here. So when it comes to touch, we have a couple of different parts to talk about. So somatosensation, as we talked about before, is anything associated with pressure, vibration, light touch, tickle, itch, temperature, pain, or proprioception. So humans have evolved to be very good at interpreting touch and the different parts of that, which is why somatosensation is associated with so many of these. And there are many different kinds. So we have nociceptors, which are responsible for sensing pain. Thermoreceptors is temperature. Proprioceptors is where your body is in space, so positioning in space. And the mechanoreceptors sense vibrations and pressure of just regular touch. And then we have specified mechanoreceptors, which are our Golgi tendon organ or GTO, which sense the stretch of tendons. And then the stretch of muscles would be our muscle spindles. So these are both important protective mechanisms so that you don't overstretch a muscle and tear it. So that's why we can also get, like, if you've ever went to the doctor's office and they tap that hammer on your knee, this is actually what is allowing you to have that reflex. And over here, we have like the free nerve endings and the different kinds of somatosensors that we have in our skin. Okay, and those were my references. And any questions?
Hey, Dylan, it's Dr. Peterson. Why don't we go back to your tongue slide and the papillae question? Because I do think we need to clarify that for everybody, both in real time and then on the recording. So can you go to the question itself?
Yes.
Okay, and so you had a lot of students that thought it could either be the taste hairs or the olfactory receptors. And then when you showed the correct answer on your slide was okay, so, but your STEM question asks which structure within the papillae was most likely affected. And I know you mentioned you had thought that you changed that, but let's just talk about that really quickly. So for those of you that said it could have been the taste hairs, the taste hairs are specialized microvilli on these specialized cells of the taste bud. So if you put that, then that's really it's a very thoughtful answer. The other choices were, Dylan, go back, let's talk about those. The olfactory receptors, the olfactory would be the sense of smell, and we're talking about the sense here of taste. So I wanted to just clarify that a little bit. I think when Dylan wrote this question, you might just talk about what were you thinking in relationship to the lingual papillae? What are they unique and special for?
Oops. Sorry about that. This light is here's- okay, yeah. So the lingual papillae are responsible for the actual functional unit that is responsible for interpreting taste. So like I said, like the taste buds. So the taste hairs, like I said, I did realize that after when I was going on to this slide. So the taste hairs is a very good answer, but the taste hairs themselves aren't necessarily responsible for taste. And I know that the other commonly chosen answer was the olfactory receptors. And as Dr. Peterson said, that's more so getting into the sense of smell. So when I was asking that question, and maybe it wasn't a fairly worded question or getting the point across, but just know that the taste buds are responsible for actually interpreting the taste, and the taste hairs are the specialized villi that are on top of the structure, if that makes sense. Hope that makes more sense to you guys.
Looks like Luke has a
Luke,
question.
yeah, go ahead, Luke.
Hi, okay, so I have a few questions. So my first one, this is kind of like a general question about like how nerves work. And so like when you like if I like touch my finger, I can feel like to the tiniest like level of touch. Is there like are there like nerves going into every last like part of the body to detect that like touch? Or is it just like one general nerve that can detect like touch around it? I don't know how to if that makes sense.
No, that's a great question. That's a very thoughtful question. So, I'm going to answer this in the simplest way that I can. So we have different nerves. So like if you were to go into like, an anatomy lab and you were ever to do like, a dissection, you're actually able to see certain nerves, and those nerves, the main nerves, have distributions that they're responsible for. So for example, like if we just take the hand, right? So the hand is innervated by, I think about like 2 or 3 nerves between the front and the back, right? So we have one nerve that's kind of responsible for like this half of my hand. We have another nerve that's responsible for this half of my hand. And then also the back, that's a different nerve. So the nerves have different territories that they cover, and then they will further branch off, but the main nerve is only one, if that makes sense. So it's not like every single branch of every nerve has like a name for it, but I would say, okay, so this one is covered by the median nerve, and this would be the ulnar nerve, and towards the back is the radial nerve. And then those give off branches. So in just kind of like a hypothetical scenario, if you were to snip a certain nerve, like if I were to snip my median nerve, I wouldn't feel anything from here to this side, right? Because all that nerve and all of its branches are cut. So they do give off branches. I hope that answers your question. It's not like there's like a single like microscopic nerve that has a different name for every single part of your skin, but they
Right.
have different territories that they're responsible for, if that makes sense.
Okay, because I was like thinking of like a tr tree trunk and then like the branches going off of it, and like each one is like innervating like a separate part of the body. But that's how I was like, okay, but that makes sense. And then like this, I hope isn't like a stupid question, but is there like a delay between like what we perceive in the world and like what's actually happening? Because we have to wait for the brain to like interpret what's happening outside of like in the world, if that makes sense. Like if we turn off the light switch and there's like no light, is there like a delay between what's like actually happening and what we're perceiving to happen?
No, so first of all, there's no such thing as stupid questions. These are really
Okay.
thoughtful questions. Um, so the transmission of neural impulses is so quick that it almost seems instant. But there are different scenarios where it would actually, there might be some sort of delay. And you could kind of think of this like in your own like daily life, for example, like if you kind of like touch something that's hot, but you don't know if it's hot or cold, it kind of takes your brain a second to be like, okay, like wait, what is this? And then you pull away. But it's a fraction of a second still, right? But if I kind of know like, okay, like I'm gonna touch my hand, it almost feels instant. It feels like there's no lag. And that's partially because of what we talked about before with the nerve impulses and the neurotransmitters. And remember how we talked about the myelin sheath. So the myelin sheath is responsible for kind of allowing that signal to kind of hopscotch. I'm just going to kind of go back. So it allows the electrical impulse to kind of do a hopscotch. So you see how we have the sheath here. So basically what the signal does is it kind of hops to the exposed parts of the axon. And that partially also is why the impulses are so fast. But on like a greater level, it's because you're also integrating a lot of different senses. So I can see that I'm touching my hand. So not only am I relying on sensation, but I can also visually, like my brain is interpreting like, okay, like I'm going to feel something right now, so that also kind of speeds it up. So I
Okay.
hope that answered your question.
Yeah. Thank you.
Dylan, I thought you did a great job handling the questions as well as presenting the material. And it was a whirlwind tour, but you did it proud. So thank you. Dr. Frank, maybe we're ready to move on to our next presenter.
We sure are. So thank you, Dylan.
Sorry.
Excellent job.
One
Now
last,
you,
sorry, I
oh,
forgot to
y-
ask this. But so, and then like I know there are some parts of the body that do like you cannot feel thing or like you can't feel pain. Like the olecranon skin on like the end of your elbow, like if you pinch it, you can't feel the pain of being pinched there. So like how does that work? And how does that differ from like pinching your like finger, if that makes sense? How does like why can't you feel pain in certain areas?
Sure. So that partially has to do with different types of skin in different parts of your body. So if you know, like your hand and your bottom of your feet, your heels of your feet have a different kind of epithelium on them, and that kind of provides like a degree of insulation so that different kinds of stimuli. So like if you kind of like drag like your finger or like a needle or something like that, you wouldn't feel the same kind of sensation if you were to drag it like on your heel versus like the top of your hand, because the epithelium is kind of insulating it. With regards to the skin on your olecranon, like on your elbow, I don't think that would be due to a deficit in innervation, but maybe more so of an epithelium. I think maybe Dr. Frank or Dr. Peterson might better be able to answer that, but that's also a very good question.
Yeah, and so Luke, you have another student that really responded very accurately, actually. So thank you, Rijva. So it's about 2-point discrimination and how close those pain or pressure receptors are in certain areas of the skin. So why don't you take a quick look at the chat and the answer that she put in, because it was really thought out very well. And then we'll go ahead and introduce Kenzie.
All right. So our second presenter this evening will give a presentation on the histology of the nerve tissue, and that will be Kenzie Stoup of Edward Via College of Osteopathic Medicine. And Kenzie is from Montgomery, Alabama. She graduated from the University of West Georgia in 2023 with a BS in chemistry and a minor in biology while playing soccer. She is currently a second-year medical student at VCOM-Auburn with an interest in OB-GYN. In her free time, she likes to play piano, play soccer, and run. Her favorite sections of anatomy are MSK and abdominal GI. So with that, I give Kenzie the floor.
Hi guys. Okay, I'm gonna go ahead and try to share my screen. Let's see. Okay, can you guys see that? Okay, great. Yeah, my name is Kenzie. As he said, this is from last year's lecture. Who gave it? You can find that on YouTube. I'm going to be doing neurology and doing the histology portion. So we can go ahead and get started. Let's see. All right, and then these are going to be the learning objectives. You guys can kind of read through those on your own. And then we can go ahead and get started with the first question. A neuron responds to a stimuli from surrounding cells and then sends a signal to other neurons or effectors. Which part of the neuron is primarily responsible for transmitting this electrical signal away from the cell body towards other cells?
Ending the poll in 3, 2, 1.
Great, choice one. And good job, the axon is correct. So we can go ahead and go to the next slide. I know Dylan talked a little bit about this already, but we can, with the histology portion, go into a little bit more detail. So here is the structure of a neuron, and you're going to have three main parts of a neuron. You're going to have your cell body right here, which is kind of the circular area with the nucleus inside. And then branching off of the cell body, you have dendrites here, and then you're going to have your axon. And the axon, as from the question stated, is going to be what the signal is going to be propagated down in order to either act on another neuron or some form of like tissue, muscle, or organ. And then the dendrites are what are going to be receiving information from usually other neurons or from the brain. And the cell body is what communicates between the dendrites and between the axon. It usually decides whether or not the signal is strong enough to continue propagating it, or if that's a type of signal that we need. And then here, this is just talking about the relationship between the axons and the dendrite, and then the dendrites. This is called a synapse here. So you'll have one cell body and it'll send an action potential or a signal down its axon, and then it will communicate with another dendrite on a different neuron, and through the synapse share neurotransmitters and then propagate that signal. And from this neuron, that will be the one that will act on your target organ or your muscle or something like that. And then this is kind of saying the same information, but if you're having a little bit of a harder time understanding the difference between dendrites and axons, This is just an analogy to visualize it more. So the dendrites are like the leaves on your tree. They can sense sunlight and they sense that signal and take it to the trunk, which is similar to the cell body, which is going to be the communicator between your leaves and your roots, or your dendrites and your axons. And then the axons are forming, you know, a network kind of communication with the soil, just as the axon here is forming communication with either the muscle or an organ or something like that. So we can go on to our next question here. And it says, which type of neuron has the functional role of transmitting voluntary motor commands from the central nervous system to the peripheral muscles?
Ending the poll in 3, 2, 1.
All right, so we kind of have a spread here. So we're going to talk about all of them. And the correct answer is going to be B, a neuron that has one axon and multiple dendrites. Sorry, I think this image is a little bit blurry, but you can see here that that's going to be a multipolar neuron. And as Dylan said, multipolar does motor, so you have two M's. And we know that a multipolar neuron is going to have lots of dendrites coming off of its cell body and only one axon. So we can look at that here. It's kind of, he talked about earlier that your multipolar is going to be your most common. So it's going to do your motor control for your skeletal muscles and for some organs. And then your bipolar neuron is the least common type of neuron, and that's going to be sensory from the retina and the olfactory. And then pseudo-unipolar, he said pseudo-sensory, so you can remember that it's going to do sensory information to your organs and glands. And then here's kind of what they look like on histology. I think there was a question about what the morphology of these different type of neurons look like. So we can see here, this is the multipolar neuron. This right here is going to be the cell body. It's kind of circular looking. And then you'll have your nucleus here. And then these smaller branches are going to be the dendrites coming off. And then here we have an axon that's long and coming off here. And then this is what's going to be doing your motor control. And then here's a picture of a bipolar neuron. This is what's going to be doing our retina and olfactory bulb. This right here is our cell body, and this is going to be the nucleus in here. And then this is going to be an axon and dendrite branching off because you have one of each. And then here's our pseudo-unipolar. You can see this is a good example. This is a cell body here with the nucleus in the middle and then some other cells surrounding it. One thing with the pseudo-unipolar is that it can be hard to see the axons branching off just because of the way the stain is cut. Sometimes axons don't stain as well, and so it can be difficult to see like a full axon. But these are some nerve fibers here running more longitudinally too. So
Kenzie,
this is just a matching question. If you guys want to like type the answer in the chat, you can do that too.
And while that's happening, Kenzie, we have some questions in the chat that probably are better answered verbally than trying to put it in the chat. So when we're done here, let's go back to the questions in the chat. Okay. And I'll help you with that. I'll help you with that.
Okay, thank you. Okay, so for the matching question in the chat here, A is going to be paired with 3 here, your pseudo-unipolar. You can see why it's called a pseudo-unipolar because it looks like it only has one branch here, but you actually have the one branch further branches into 2. So that's what the pseudo is for. And then B is going to be our bipolar here. You have one axon and one dendrite kind of going perpendicular to each other. And then multipolar is going to be here with our multiple dendrites. And then we can go ahead and talk about those questions. I can go back to the slide.
Right, and so the question was, why does a multipolar neuron have so many dendrites if its goal is to innervate and ultimately send signals to 1 plus muscles or 1 plus other neuron in a circuit. So if you want to jump in, and then Dr. Frank and I can kind of throw in our 2 cents worth. How about that?
Okay, I don't know if I have a perfect answer on this. My my thought would be that it needs to receive a lot of sensory information to go and act on an entire muscle. I don't know if you guys have a better, I'm not sure on the exact reason on why there's multiple dendrites.
Right, so and this is complicated, and we would not ask you this on the exam. It's a little bit beyond the scope of the local competition, but there's 2 different ways where a cell body brings together the total signals it's receiving. So it can do it through a summation process, so just enough axons releasing their neurotransmitter onto multiple dendrites of a single axon that it allows an action potential to be generated and start transmitting down an axon, or it can be what's called a temporal culmination. So a lot of neurotransmitter released very, very quickly onto 1 or 2 dendrites can also generate that action potential. And that's kind of how the cell body, in what Kenzie was describing, determines if the signal should be moved on or not. So hopefully that at least gets to the heart of that question. Dr. Frank, you want to add anything?
I was just going to piggyback with you. Some of the neurons that are synapsing on the dendrite are actually going to be inhibitory. And so we have some that are going to shut off the transmission to the next neuron. There are others that are excitatory and are going to excite the next neuron. So we have to have the opportunity for area for both, so that we can better regulate and integrate the nervous system. So
Was there any other questions on this slide as well?
No. So, and I
No.
think, yeah, I think definitely move on, but you did a great
Okay,
job.
perfect. Thank you.
yeah, excellent questions, everyone.
All right, so this is our next question. It says that nerves are divided into multiple organizational layers, with each layer being surrounded by connective tissue sheaths. So which of the following answer choices correctly pairs a connective tissue sheath with its contents?
We'll end the poll in 3, 2, 1.
All right, so a little bit between choice 1 and 2, so we can go ahead and talk about that. Let's see. All right, so the perineurium, choice A, surrounds fascicles, so that is correct. And then going on to the slide here, this is going to be showing a nerve and all the different parts, so we can walk through it. We can start on the outer layer here. So this is the nerve as a whole, and the epineurium is going to be surrounding the entire nerve. And so then this is also a cross section of the nerve. So if you were to take your arm and like cut it like this, you're looking up at the nerve like that. So inside of the nerve, you have different compartments, and these are called fascicles right here, these big circles. And those are going to be surrounded by the perineurium here, so around that. And then within the fascicle itself, you're going to have the individual axons. And these individual axons are going to be surrounded by the endoneurium. And I think it's helpful to understand what these words mean so that you can easily like remember if you get this on a test question or something. So if you look at the prefix epi, that usually means superficial if you think like the epidermis of your skin. And then endo usually means within or internal. So I like to remember endo has an N in it and internal has an N in it, or within has an N in it. So the way I always remember this is that the perineurium or peri is sandwiched between your two E's. So it's going to go epineurium, and epi means outer. So that's going to be first around your entire nerve. And then your perineurium, and then your endoneurium is on the inside. And then the reason that we have these sheaths in the first place is to protect the nerves. And then that helps propagate signals better. And each axon is sending its own signal. So you want to protect that. And then also here, the epineurium contains some blood vessels and things like that. And then here's a histology picture. So again, this is a cross section of a nerve. So you're cutting it in half. And we can see here that this is our entire nerve, and we see the epineurium on the outside here. And then the fascicles are here, and the perineurium is surrounding that. And then this is a zoomed up version, and you can see these little dots are the individual axons. And the endoneurium is this darker pink that's surrounding each one of these little purple circles. So then if we go here, question number 4, it says, which of the following represents a bipolar neuron? So we can go ahead and just look at the histology.
Ending the poll in 3, 2, 1.
Job, choice B, that's correct. So again, here we have the multipolar neuron. You can see all these dendrites branching off the cell body. And then the bipolar, you have just 2. And then here you can see the cell body, the pseudo-unipolar, but again, it's hard to see that axon coming off sometimes just with the way the stain is. So this is just kind of a side about looking at axons versus dendrites on histology. It can be a little bit difficult to tell what is what sometimes. But the axons are usually a little bit paler. You can see in this image here, it is kind of fading out. And then again, here the axon is a little bit paler. And then also axons tend to keep their width for most of it. So they stay kind of fat the whole way through. You can see here that this is a little bit larger. And then here, this is about the same width. And then dendrites are typically darker in color. And then they're going to taper. They're going to start fat, and then they're going to taper off to a point at the end. So if you're having a hard time differentiating, that can be helpful. All right, and this is our last question. So for this one, it's probably going to be easier to go ahead and just type your answers in order in the chat, and then we can talk about them.
We have A is 4.
Yes, that's correct.
1 is C.
1 is C. Yep, that's correct.
B is 2.
Yep.
Okay.
And that leaves D. So we can go ahead and look here. These are the answers. Again, yeah, this is an entire nerve section here, and within the nerve, these are individual fascicles. And we know that the fascicles are composed of individual axons here, and those are those little dots. So 2 here, this is the fascicle, and then the perineurium is going to be around that. And our epineurium is the thickest, and it's covering the entire nerve all the way around. And then we can just take this section and zoom it in, and we can see our axons, and then the dark blue around these individual dots, that's our endoneurium. And then this is the same slide from earlier, just with that cartoon, if you're having a difficult time visualizing that. And then that is all I have. Do you guys have any questions for me?
We go back to that previous slide. For the arrow of number 2, was it
Yes.
pointing to a fascicle or a specific endoneurium?
Yes, this is pointing to a fascicle here. You wouldn't be able to see the endoneurium if it was, unless it was zoomed in, kind of like this one here, because number 3 here is pointing to the endoneurium. I guess on the question, I should have specified if I wanted the whole structure or the sheath specifically, but yeah, I was getting at this being the entire fascicle.
And we would normally do that. We would put a circle. If this was a test question on the local competition, we'd have a whole red circle around that or a bracket or something so that you would better know what we were asking you to identify. The other thing, and I know that some of you are just joining us for the first time, but earlier on in one of the very first tutoring sessions, we mentioned that there are special stains that are sometimes used on different types of tissue. And typically when we see anything that is stained a bright blue like that, they're using a special stain that will highlight the connective tissue. So all of these different wrappings that Kenzie was describing to all of you, that is fibrous, thick connective tissue on the outside at number 1. And you can see that it's getting thinner and more fragile as we move from the outside to the inside. So you can barely make out around the endoneurium over here on the right-hand side around those individual axons. It's a very, very thin blue line, which suggests it's a very, very thin fibrous connective tissue sheath around it. And then I'll just for completion's sake, Kenzie did a really good job of that. But on this blown up view, highly magnified, the dot right in the center is the axon itself, and all of that white material around that dark colored dot, that is the myelin, the lipid insulating sheath that Dylan talked about. So hopefully that kind of is bringing a lot of these 2 presentations together for all of you, and that's great.
Are there any other questions?
I know that there were a few questions along the way asking about the significance of a unipolar neuron versus pseudo-unipolar versus bipolar. And Dr. Peters and I were just saying that, first of all, there's no unipolar neurons in I think squids, kind of notorious for having them, but not in humans. But it was just more to show the difference between unipolar, pseudo-unipolar, bipolar, and multipolar. So not to get more in depth than that. We're just using it for reference. But did you have anything else that you wanted to add to that, Kenzie?
I don't think so. No, I think, yeah, the unipolar is just to understand where the like word pseudo unipolar comes from. But yeah, other than that, no, I don't have anything else to add.
Another question, and it is difficult. I understand this can be frustrating for those of you that are just starting to look at histology slides and try to figure out what is getting stained and what you're looking at. A question got asked, how can you identify the dendrites and axons in a histology stain? I failed to identify the other structures due to other cells. So I think it just takes, and then I'll let Dr. Frank and Kenzie jump in. I think it just takes looking at a lot of these different cell types side by side by side. And sometimes we show you that in the tissue sample and sometimes we do not. A really good way to study histology is just go to Google and then up in the right-hand corner, click images only. And then for your Boolean terms in the toolbar, just say multipolar neurons and you'll see hundreds of multipolar neurons side by side. And you can start to get a better sense of what Kenzie was describing about dendrites being shorter and more numerous, oftentimes staining darker, and the axon, which may be then out of the tissue section and how it wove its way through that tissue sample. It should be the longest one. It should be lighter staining, but you may not see all of it because, again, this was in a three-dimensional tissue sample, and we just cut a little slice off of it. That's the hard part of histology, I think.
So Deepthi had a question. So just to confirm on the histological stain, the white areas are the myelin and the blue areas are the neurons, or is it the opposite?
So the white area is the myelin and then, or the myelin, excuse me. And then yes, these these purple dots in here, those are going to be the axons of the neurons, if that makes sense.
And as Dr. Peterson said, the coverings around our fibrous tissue that make up the epineurium, perineurium, and endoneurium.
Yes.
Kenzie, thank
Okay.
you so much. That was awesome.
Awesome
They-
job.
Yeah, they learned a lot. I mean, I know they did from you tonight. So thank you.
Thank you.
Yes. Thank you for coming back. Okay. Well, that leads us to our last presentation, and this will be on embryology, specifically neurulation. And we have with us this evening Emilie Savaikie from Pacific Northwest University College of Osteopathic Medicine. So Emilie is from Auburn, Washington, and graduated from the University of Washington with a Bachelor of Arts in Political Science. She is currently a second-year medical student at PNWU COM with interest in OB-GYN and emergency medicine. Emilie previously worked as a CNA in the ED and school health settings, as well as volunteering with organizations such as NILMDTS. And Emilie, you need to explain what that is to this group, either beginning or the end. I think it's absolutely fascinating, and I'd love to hear a little bit more about it. I obviously have looked on the internet, so I think that would be great to share what that is and the work that you did there. I just want to say, lastly, in her free time, if you have any, um, she enjoys bike rides and playing Minecraft with her kids. So, with that, um, Emilie, it's all yours.
All right. Thank you so much. So let me share my screen. PowerPoint and share. Okay, hopefully that's coming across. And yeah, I will share my stories about Now Lay Me Down to Sleep at the end. But we're going to cover neurulation. It's a key part of embryology and the growing fetus in embryo. Sorry. Some of the lecture objectives. So we want to define neurulation and its significance in embryonic and human development. Describe the formation of the neural plate and the transformation into the neural tube, discuss the causes and effects of neural tube defects, and define the origin and migration of neural crest cells. So we're going to go back to the last tutoring session, beginning in week 3. So we have the formation of, we have a epiblast and a hypoblast and our bilaminar disc. And so we get a primitive groove that forms to create that primitive streak. And you have these these epiblast cells will migrate and will start forming a trilaminar disc. So you're going to have 3 layers of cells. So you start with your endoderm, your mesoderm, and your ectoderm, forming these layers of your trilaminar disc. And that should complete around or so within week 3. And so just to make sure everyone's listening, which layer of the trilaminar disc forms the central nervous system? Okay.
Give 3, 2, 1.
Oh, excellent. It is the ectoderm. But another structure I want to talk about before we get into the full part of neurulation is this notochord. Um, so mesoderm cells, a part of the trilaminar disc, form the solid rod known as the notochord, and it influences how the embryo folds onto itself and submits or produces signaling molecules such as, and I love how people who discover a molecule get to name it. So this one's called sonic hedgehog, is one of the main molecules released by the notochord, and it guides the tissue differentiation. And then somewhere in the middle of week 3, we have the notochord will start stimulating this central part of the ectoderm cells to form this neural plate. And this thickening tissue um, will start growing, and then you have these neural plate border cells, which will form the neural crest coming up. And they'll fold onto itself. And it's really hard to kind of imagine trying to be in this 2D image, but you get this proliferation of neural plate cells and it keeps growing. And you have these edges that start to come together with this inward folding, and it forms what we'll call the neural groove. And then to form the neural tube, the folds migrate medially and they will fuse, and it pinches off and forms this neural tube. The border cells that got pinched off are your neural crest cells, and they form parts of your peripheral nervous system, among some other things we'll get into. And then your notochord, its job is done, so it eventually disintegrates, and it still survives to this day in your body as the nucleus pulposus of your intravertebral discs. So when closing of the neural tube, the tube closes in both directions, middle, and then closing toward the head and the tail ends like this two-way zipper. So the head end, we call the cranial neuropore, it seals around day 25, and the tail end, the caudal neuropore, caudal meaning tail, and then I said cranial neuropore, cranial meaning head, seals around day 28. If either end doesn't close properly, it can lead to neural tube defects, and we'll get to that in a second. And this shows how early and crucial neurulation is for the healthy development of the brain and spinal cord. So there's some failures with neural tube closure. So what happens if we don't close? So we call these neural tube defects. On the cranial neuropore, and if it fails to close, you get something called a condition called ancephaly. There's some other conditions that can happen, but most commonly is ancephaly, where the brain doesn't continue to form. On the caudal end, you have a condition called spina bifida if that caudal neuropore fails to close. Some of the causes for this, low folate or folic acid or vitamin B9 prior to pregnancy. Genetics often play a role, and the mother's metabolism, such as issues with gestational diabetes, which can happen to pregnant people when they get pregnant. So those neural crest cells, they form a lot of other structures. So they start to migrate all over the body. And in the head, they form your facial bones, cranial nerves, and the connective tissues, parts of your head. Along the spinal axis, you get your peripheral nervous system, so your sensory and autonomic nerves, melanocytes, and chromaffin cells of the adrenal medulla. Just as a quick note, chromaffin cells, they're part of your adrenal gland, and they release the epinephrine or epinephrine. And it's activated by your sympathetic nervous system or your fight or flight system. And your parafollicular cells of the thyroid, they release a hormone called calcitonin, which lowers the blood calcium by stimulating osteoblasts to form new bone and inhibiting osteoclasts from breaking it down. So just a summary of neurulation. It does begin during week 3. The notochord forms from the mesoderm and releases signaling molecules to guide development. The medial ectoderm thickens to form the neural plate. The neural plate then folds to form the neural groove, and the neural groove fuses to form the neural tube. And those neural crest cells migrate through the body, forming the PNS and other tissues. The tube closure completes day 25 for the head and around day 28 for the tail, and forms the central nervous system. So we'll do our second practice question. So the notochord is derived from which embryonic layer? And sorry, I can go back to previous slides if I'm going too fast. I apologize.
That's okay. We'll end the poll in 3, 2, 1.
All right. Oops, that should have been mesoderm. I am so sorry I highlighted the wrong thing, but you're right. Choice 3 is correct. So the next question, zygomatic bone, otherwise known as a cheekbone, derives from which embryonic structure? Paraxial mesoderm, neural crest cells, or neural tube cells?
End in 3, 2, 1.
Excellent. Neural crest cells is right. Let me advance the slide. There we go. Fourth question. We just got a couple more here. Notochord derives. Sorry, that was a repeat here. Neural crest cells are formed from which? I'm sorry, a couple plates. Oh, sorry, yeah. Which of the following structures? The neural plate border cells, the neural groove border cells, or the ventral neural tube cells?
Ending the poll in 3, 2, 1.
All right, we got close to choice 1 and choice A and B, but we are the neural plate border cells. And we can go back to that slide. Just remember that plate leads to groove, leads to tube. All right. Cells that myelinate only the peripheral nervous system axons, known as Schwann cells, come from which embryonic structure? Neural crest cells, neural tube cells, or notochord cells? And this is the last question I have.
Ending the poll in 3, 2, 1.
All right, you guys are right. Neural crest cells form the Schwann cells, which are part of the peripheral nervous system. So that's what I have here are my references. And I'm ready to open up some questions, and I can talk about now laying me down to sleep a little bit after we tackle some questions.
Dr. Peterson, I wanted to point this to you. Um, uh, Tasneem has asked if we're going to test on in embryology. And because I go to Learning Target and it just says anatomy and histology. So could you clarify that for everyone? Not sure where they're referencing, but
Right. So again, I know some of you are joining us for the first time, but on the website, under the resources tab, you will find learning objectives for the anatomical sciences. And so for Anato-Bee, we are going to test you directly from the local competition learning objectives. Those are the ones in the very first column on the table for the following three different types of sciences that are under the umbrella term anatomical sciences. So be gross anatomy number 1, histology number 2, and embryology number 3. And then Dr. Frank, I saw that there were quite a few students commenting on the PowerPoint slides and trying to take notes from those. This year, for the first time ever, yay, we are posting the presenter's PowerPoint slides in the Google Classroom because that's private and these are sort of proprietary PowerPoint presentations. So we didn't want them to be posted with the YouTube videos. So now you have several different resources. You have YouTube videos made from PowerPoint slides from 2 previous years. Then you have this year's YouTube video being posted as we move through the tutoring sessions. And you could take screenshots of the different PowerPoint slides as they came up in the YouTube video. But as one of the students mentioned, and again, I'll mention it, the PowerPoints in and of themselves are getting posted into the Anato-Bee Google Classroom. So I hope that helps.
are there
So
other
how about
questions?
questions for Emilie?
Well, I want to know more about Now I Lay Me Down to Sleep.
Sure. So now laying me down to sleep. Before I went to medical school, I was a professional photographer, but if you want to volunteer with this organization, you don't have to be a professional, but what we do is we'll take remembrance photos for families who lost their baby around the time of birth. So we'll come in, we'll take photos that kind of resemble the photos that you would have in life. Sometimes you have the froggy baby poses where you have the baby falling asleep with the hands up, and some of the cute poses that we have. A lot of the parents are missing those when their baby passes around the time of birth. So we give them an opportunity to get those photos and just to say a little goodbye. So it's very fulfilling. I wish I had more time for it now. But I thought that was really fulfilling, and I've done it for many years, and I plan on continuing it after I have some more free time.
I just think that's fascinating. Thank you for that. That as um, a parent that lost a child, I wish we would have had that. So much appreciated. But getting back to embryology of the nervous system, we do have a question. Where do the neural crest cells come from again? And this is from Balan.
Yeah. Let's go back to um, so when we start forming this neural plate, where the ectoderm starts thickening up just a little bit, around the edges, we have these neural plate border cells, which divide it from the ectoderm and the neural plate. And as this pinches up, you're just gonna have to imagine this evagination forms as the neural plate thickens to form a neural groove and just kind of folds into this V shape. And then to form the tube, it just pinches off at the end And then you have these cells that just kind of pinch right off, and now they are free to migrate, and they don't stay there anymore. They go all over the body. And there are certain chemicals and signals and molecules on cells that keep cells where they are, and those cells no longer express some of those molecules, so they are free to roam and follow other signals and develop other structures.
So just following up, Rashab asked, what are the three red circles in the diagram? And then Karav has a question.
Oh, sure. I'm gonna assume this is gonna be your paraxial mesoderm on these two that's adjacent or on both sides of the neural groove, and then below that's the the notochord that's sending out those signals to aid in the development of the neural tube.
And I'll just mention, Emily, that's exactly correct. I don't want anybody to get too excited or stressed. You don't know anything yet about the mesoderm specialization, the paraaxial, the intermediate, and the lateral plate mesoderm, but you will when we talk about how the limbs develop from the little embryo, basically core trunk. So paraaxial mesoderm is coming. And it's just amazing how much specialization occurs in such a short period of time, as well as to think there are cells that are kind of sliding around, not only in the embryo body, but in the human adult body as well. I find that kind of comforting at the end of the day to think, hey, even though when I lay down and go to sleep, there are still cells inside my body that are moving around. So it's kind of a cool concept. All right, so Kairab, I think we're ready
Yeah.
for your question.
First of all, I want to thank you for that presentation. That was awesome. So coming to my question, I looked at my previous year notes and I've written that like when a person has spinal bifida, they have an overlying tuft of hair. I just wanted to ask you why that is.
Um, I think some people probably will answer that better than I can, but there oftentimes there's this condition called spina bifida occulta, which isn't the full so spina bifida is on this huge continuum. It could be just a small amount of attachment with a tuft of hair or a discoloration that's on the base of the spine, around the sacral area where it meets, or it can be a complete blowout where you have these meninges that are opened out with parts of the spinal cord exiting the body cavity. And it relates to all sorts of different levels of pathology or disability from that, but it can vary quite a bit.
Okay.
And I'll just follow up on that, Emily, you did a great job of that. So on this continuum, what we really are thinking about is from the notochord, did the vertebrae form around that developing neural tube, which is going to become the spinal cord and the brain at the cranial end? So we need the vertebrae, the bony portion, to form around that and protect the spinal cord. But in addition, and that comes from the paraaxial mesoderm. But in addition, you need the skin to close over that as well.
Mhm.
And there's some signaling molecules that have to happen for the vertebrae to close. When they don't close, that signaling just keeps spilling out more signaling molecules, which scientists believe sort of overstimulates the ectoderm, the skin, to just grow more hair, have more hair follicles develop there.
Okay, thank you. I think that answered my question.
And there are different severities of spina bifida. I mean, this is the worst case scenario, but in spina bifida occulta, the only sign you may have that there is an incomplete fusion of the neural arch of the vertebra is the tuft of hair on the skin. So obviously that would lead the physician to ask more questions and possibly do some testing. So we also have, um, uh, can you explain anencephaly again?
Yeah, absolutely.
Balan?
Ancephaly. So if we go back to this crazy drawing here of this neural tube. So on this cranial end, if you don't have that closure, you can have a symptom called, or a condition called anencephaly, where the brain doesn't form. So without a brain or without the top of the skull, you have um, nervous tissue mixing in with amniotic fluid, which is very toxic to nervous tissue and will kill this off. Also note, this is around week 3 to week 4. This is about a week and a half of things happening. Most women don't know they're pregnant by this point. This is week 4. This is very early. So the fate of the baby could already been decided before someone knows they're pregnant. But anencephaly, just in brief, that's when the brain and the head, parts of the head fail to develop. And I didn't put pictures on there, just I didn't think they were um, great for this audience, but definitely with you see it on the other end with the spina bifida. You just have this open end, and in this case, you don't have spinal cord exposed to amniotic fluid, but because it is protected. So, but you and you can get different levels of disability based off of that. But anencephaly is typically not compatible with life.
I know this is above our area here, but Rashab asked, how do you treat spina bifida? And obviously it depends on the severity of the spina bifida. And so Emily, did you want to add anything?
From what I remember, yeah, it depends on severity. It may be you don't need to do anything, that tuft of hair. You can get tethering of the spinal cord with signs like that, where just part of the phylum terminale, I believe, that the last little string that hangs off of there just attaches to, I can't remember where it attaches, but it ends up locking that spinal cord in a little bit. Could be one of the more milder forms of it, but when you have a larger exposure, you want to be able to try to protect that from the environment, be able to tuck a lot of this back in, close it up, and to protect that from further damage. And then it's for more severe cases, it's going to involve disability aids and physical therapy.
And I'll just add that this is depending on the severity, again, the amount of exposure. This is all detectable through ultrasonography. So again, this is not the same kind of scenario as anencephaly. Again, not compatible with life. This is the embryo continues to develop to a fetus. A woman goes for her mid-gestational period appointment. The physician will see this on an ultrasound, and they can start to plan for what the surgical treatment will be, either just after birth and now. And this is again, this is crazy cool, is that they are doing transvaginal surgery. So they go in even before the fetus is born, and they surgically correct it to, as Emilie suggested, to minimize the exposure of that spinal cord and any kind of actual loss of cell viability at the caudal end of the spinal cord. So it's just minimizing the amount of damage done to the cord.
I haven't seen it yet, but that is absolutely I've heard of it. I haven't actually seen the procedure, but just absolutely amazing that you can perform surgeries on embryos in utero. So, and for the most part, it will mitigate or soften the damage if it's pretty severe. It's not going to prevent it completely. If you have that much damage from failure of closing that caudal neuropore, there's going to be some disability. The level is just going to be based on how much time it's exposed and how it's repaired.
Okay, do we have further questions? Again, really good questions this evening from everyone.
All right, Dr. Frank, I think we finished a little early, but I think we all learned a lot tonight. So really proud of all of you. Hope you enjoyed this. Again, the recordings will go up on our YouTube channel, which you can access through our website sometime tomorrow, as well as we'll get the PowerPoints posted in the Google Classroom. And we look forward to seeing you in 2 weeks.
Bye, everyone. It was a pleasure.
Good
Thank
evening.
you.
Take care.